This video is going to cover section 3.3 and 3.4. Again, we're continuing to look at cell structure and function, and here we're going to look at the cytoskeleton of the cell. The proteins of the cytoskeleton, they interconnect and they extend right from the nucleus to the plasma membrane. And the cytoskeleton, it allows eukaryotic cells to maintain their shape and for the organelles to move. There's three compounds of the cytoskeleton network, and they are actin filaments, the intermediate filaments, and the microtubules. And here you have, it looks like electron micrographs of those, although it's a little difficult to pick them out and actually see what shape they are. So we'll look at them in more detail here. The actin filaments, they're long and extremely thin in the area of about 7 nanometers in diameter. They are flexible fibers that occur as bundles or mesh-like networks. And each filament, it contains 2 chains of globular actin monomers, and those are twisted about each other in a helix kind of a shape. And you can see that from the diagram here. In the diagram A there, from left to right, you can see that the fibroblasts in animal tissue, they contain actin filaments, and the drawing then in the middle, it shows that actin filaments, they're composed of a twisted double chain of actin subunits. So the giant cells of the green algae Chara, they rely on actin filaments to move organelles from one end of the cell to the other, and that would be true of many cells actually. The structure and the movement then, so actin filaments, they play a structural role by forming dense complex webs under the surface of the plasma membrane. They're also in microvilli, which project from intestinal cells, and pseudopods, which are extensions that allow some cells to move, for example, the amoeba. Actin filaments, they play a role in movement by associating with what are referred to as motor molecules, and these are proteins that attach, detach, and reattach farther along the actin filament. And this is especially true in muscle cells, and myosin is a motor molecule, and the head of the myosin interacts with ATP, thus giving it the energy, and actin, and the tail interacts with the membrane. So myosin then, it pulls actin filaments along and derives the energy to do so from ATP molecules. The intermediate filaments then, they're intermediate in size between the actin filaments and the microtubules, about 8 to 10 nanometers in diameter. And this is a rope-like assembly of fibrous polypeptides. And again, polypeptides are chains of amino acids. And you can see in the diagram here that fibroblasts in animal tissue, they contain intermediate filaments, and the drawing shows the fibrous proteins that account for the rope-like structure of these intermediate filaments. Hair is actually strengthened by the presence of intermediate filaments. That brings us to the microtubules. So these are very small, hollow cylinder-like structures, and they're in the area of 29 nanometers in diameter, and about 0.2 to 25 micrometers in length. So these are composed of 13 rows of alpha and beta tubulin dimers surrounding an empty core. So those terms just refer to the structure of it. And again, we have an electron micrograph of it by the looks of it there. And so the fibroblasts in animal tissue contain microtubules, and the drawing in the middle then, it shows that microtubules are hollow tubes composed of tubulin subunits. The skin cells of a chameleon relies on microtubules to move pigment granules around so that they can take on the color of their environment. So if you ever wondered, how a chameleon does that, it's through this mechanism actually. The microtubules allow them to move pigment around their cells, thus changing their color quite quickly too, actually. So the microtubule assembly then, in most eukaryotic cells, the centrosome, it contains a microtubule organizing center, and that regulates microtubule assembly. So microtubules, they radiate from the centrosome, and tubules help maintain the shape of the cell, and they act as tracks that organelles can move along with the aid of the motor molecules, as in kinesin and dynein. Now, before cell division, microtubules disassemble and then reassemble into a spindle, which attaches to chromosomes for proper distribution and participates in the dividing of the cell. So as far as the movement of the chromosomes, then that relies on the microtubules as well. So then after cell division, the spindle disassembles and microtubules reassemble into their former arrangement. Centrioles then, in animal cells, a centrosome contains two centrioles. And these are oriented at right angles to each other, actually. And the centrioles, they're short cylinders of microtubules in a 9+0 pattern of triplets. So a ring of 9 sets of microtubule triplets. Centrioles, they replicate before cell division, and each pair then becomes part of a centrosome. And the centrosomes then move apart at cell division. Cilia and flagella, these are hair-like extensions that move. And so they're extensions from the cell. They provide movement for some cells, and they both consist of membrane-bound cylinders composed of a 9+2 pattern of microtubules. So 9 microtubule doublets in a circle around 2 central microtubules. So cilia and flagella, they move when the microtubule doublets move past each other. And the protein dynein acts as a motor molecule in this case. And you can see it's illustrated here in the diagram. So the movement of the arms then allows for movement of cilia and flagella. Once again, it requires ATP to provide the energy in this case. So that looks at those three structures of the cytoskeleton, which are the actin filaments, the intermediate filaments, and the microtubules. So now we'll look at the plasma membrane structure and function in a little bit more detail. So the plasma membrane, it regulates the entrance and exit of substances from the cell to help maintain homeostasis, of course, what else? So the fluid mosaic model of the plasma membrane then, it is described as such with phospholipids forming a bilayer, and the hydrophilic polar heads, they face inside the cell and outside of the cell, and the hydrophobic tails, they face each other, which forms the middle part of the cell membrane. So peripheral proteins are partially embedded on one side of the membrane. There are also integral proteins that span the entire membrane, and they may protrude into one or both sides, and they move laterally, which means from side to side. So steroids, glycolipids, and glycoproteins are also present within the cell membrane. So here's a diagrammatic representation of this, and it shows it in a fair amount of detail here. So you can see the peripheral proteins, some integral proteins, filaments of the cytoskeleton, etc. So the functions of the membrane proteins, the plasma membranes of different cell types, they have unique combinations of proteins. So generally, the peripheral proteins, they play a structural role by helping to stabilize and shape the plasma membrane. The integral proteins, which span the membrane itself, they determine a membrane's specific function. So these would be examples of those. You could have channel proteins, carrier proteins, cell recognition proteins, receptor proteins, and also enzymatic proteins. So the channel proteins, they're involved in the passage of molecules through the cell membrane by forming a channel. Carrier proteins, they're involved in the passage of molecules through the cell membrane by combining with the substance. Then we have cell recognition proteins, they're glycoproteins involved in cell recognition of pathogens. Then we have receptor proteins which bind specific molecules which results in a cell response. And then finally, enzymatic proteins, they catalyze cell reactions. So some of the reactions that take place within the cell, they require the enzymatic proteins in order for them to occur. So that will end our discussion then on the cell membrane and the cytoskeleton. And so here's the summary of some of the key terms and concepts that we just talked about.